So coming to hematopoiesis. So hematopoiesis, what does it mean by hematopoiesis? Hematopoiesis means it is the production of cells. It is primarily starting from the hematopoietic stem cell. There is a stem cell called as hematopoietic stem cell. From the stem cell, all different types of cells are produced. So basically, they are classified into two things. One is the myeloid series, that is the myeloid group of cells, myeloid series, and another group is lymphoid series. Lymphoid series. This lymphoid series will include the T cells, B cells, and the NK cells. So they basically include the T cells, B cells, and NK cells, natural killer cells. So they form the lymphoid series. Whereas the myeloid series is further subdivided. They are further subdivided into GM-CFU and EM-CFU. What is the CFU? It is nothing but the colony forming units. The CFU stands for colony forming units. So this GM-CFU means it is the granulocyte monocyte CFU. Granulocyte and monocyte. And this EM-CFU means it is the erythrocyte and the megakaryocytes. So this is the basic classification. And all of us know that granulocytes, there are three different types of granulocytes. From this granulocytes CFU, they will form three different types of cells. That is the neutrophils, which is granulated, eosinophil, and the basophils. So these three cells are called as granulated cells or granule cells and basophil. Whereas this monocyte group will give it rise to the monocytes in the blood. Then erythrocytes, they are the ones which is going to form the RBCs, that is the red blood cells in the body, which is very, very important physiologically. They form the erythrocytes. They are formed from the erythrocytes. Then coming to this megakaryocytes, what do they form? They form something called as the platelets. They form the platelets in the blood. So these are the basic hematopoietic stem cells and their further divisions. Now coming to the process of erythropoiesis. Erythropoiesis means it is the process of formation of RBCs. So RBC will be our end product. That is simple because that is what we are going to form in this. Then RBC starts from the pro-erythroblast. That is the most precursor stage of RBC. From the pro-erythroblast, they undergo various different stages and finally they form the mature RBCs. They undergo the stages like early normoblast. They form early normoblast. Then intermediate normoblast. If early is there, then there should be some late. So the third one is late normoblast. In between these two, we have something called as intermediate. So the intermediate in between these two, that is called intermediate normoblast. So these are the basic classification of this. Then from the late normoblast, they go into a particular stage called as the reticulocyte. So this is how the RBCs are formed. This intermediate normoblast is also called as polychromatophilic normoblast because initially they will be basophilic and they will start changing into eosinophilic. Here both are present. That's why it is called polychromatophilic, multicolored. Now we have to know some processes in this steps of RBC formation. First, in the pro-erythroblast stage, what is there? The mechanism for hemoglobin formation, the elements required for hemoglobin formation, everything is present. But they will be visible only with the help of electron microscope. It is not visible through normal microscope. It is visible through electron microscope. And the intermediate normoblast is the one where in the light microscopy, hemoglobin starts to appear. So in light microscopy, hemoglobin starts to appear only in the intermediate normoblast states. Then this reticulocyte, which is just one step behind the RBC, is also very, very important. Why it is important is this is the cell which is released into the blood, and after 2 to 3 days, they are converted into RBC, and their count is usually increased during a hemorrhage. Because during an hemorrhage, more and more RBCs are going out of the body, and finally they have to be replenished. So more reticulocytes will come into the circulation. And this reticulocyte count is also essential for one more reason. It is helpful in assessing the treatment. Suppose as iron therapy is given for a patient, we have to check for the hemoglobin levels. The hemoglobin level starts to increase after a delayed period of 7 to 14 days. But before that, I should know whether the therapy is working. For that, we can take help of the reticulocyte count. So it is used to assess the therapy. So these are all the functions of the reticulocyte. Then finally, we are formed with our RBC. This RBC, they are usually 5.5 millions count is there, and they are biconcave in shape. They are biconcave cells. All of us would have observed it in the microscope, and they are biconcave in shape. Then it has two important proteins, that is the structural membranal proteins. They are called as the ankyrin and spectrin. There are several proteins, but two of them are very, very important. Among this, you can see here the spectrin is spread over a wide range of area. The black ones here are the spectrin, and the colored ones here are the ankyrin. Tell me which is the most important or the most dominant one which is involved in defects. Like most commonly, which is involved in defect. Most commonly, if they ask, the answer is ankyrin. Most common defect is seen in the protein ankyrin. But if the question is which among these two indicates the severity of the disease, spectrin, just remember S for S, spectrin is the one which is essential for the severity of the disease. Then spectrin is the one which is responsible for the deformability of the RBC. It is the deformability. The RBC can squeeze in through minute capillaries. That deformability property is given with the help of spectrin. So these are the two important proteins. So coming to the functions of RBCs, RBC has one important molecule inside it which is called as the hemoglobin. So whatever is the function of hemoglobin, like carrying oxygen and delivering throwing out carbon dioxide, all this are the functions of RBC also. Not only that, they also have the blood group antigens. Whatever blood group we are going to study, the antigens for them are present on the surface of the RBC. So both are the functions of the RBC. Now coming to a discussion of the hemoglobin. The hemoglobin is the one which is the oxygen carrying pigment. It is the one which carries the oxygen, and it is a tertiary protein. It is a tertiary protein, like more mature protein, and it primarily consists of iron porphyrin ring. It consists of iron porphyrin ring. How the hemoglobin is formed? Hemoglobin is primarily formed by two succinyl CoA and the glycine. Whenever two succinyl CoA molecules and the glycines are interacting with each other, they will form something of a ring that is called a spiral ring. This spiral ring is formed. This spiral ring is one ring. If there are four spiral rings which are coming together and they will form they will together form a protoporphyrin. This protoporphyrin is the one which is the formation of four spiral rings. Then finally, when Fe is added, that is ferrous is added, then it becomes the heme. So for forming a hemoglobin, we need two products. One is the heme and the globin part. Globin is the one which is a protein part. Till now, we have formed our heme from the primary products of succinyl CoA and glycine. So finally, heme is there, and if the globin protein is available, they together will form the hemoglobin molecule. So heme is the iron containing pigment and protein part is the globin. In an adult hemoglobin, there are four chains. These four chains are called as two alpha chains and two beta chains. Whereas the hemoglobin F, that is the fetal hemoglobin, will have two alpha and two gamma chains. And we know that the gamma chains is responsible for the increased affinity of the fetal hemoglobin. That we have seen it in the respiratory system itself. This four chains will have two alpha and two beta, and all four of them can combine with one molecule of oxygen. So in total, how many molecules of oxygen can be carried by hemoglobin? Four molecules of oxygen can be carried by one hemoglobin molecule. Then coming to two disorders of hemoglobin, wherein there is a replacement of amino acid. The first one is called as sickle cell anemia. Sickle cell anemia. In sickle cell anemia, what happens is the amino acid valine is replaced by glutamate in the beta chain. So valine replaced by glutamate is the answer for sickle cell anemia. Valine is replaced by glutamic acid. And there is another form of hemoglobin which is called as HBE. It is also a single point mutation where glutamic acid itself is replaced by lysine. the beta chain glutamic acid is replaced by lysine. So these are the 2-point mutations which is creating this kind of sickle cell anemia and the HBE variant. So coming to the hemoglobin synthesis. Now we have all these byproducts, but there are some essential products or the minerals and proteins which is required for the synthesis of hemoglobin. First thing is it needs a lot of nutrients. The subject should have a lot of nutrients. Followed by that the protein globin should be there, and one very important mineral, that is the iron. The iron mineral should be there. If there is a deficiency of iron, all of us know India's most common anemia causes iron deficiency anemia, because the mineral iron is very very essential component for the formation of hemoglobin. Then vitamins that are required are vitamin C, B12 and folic acid. And we know that this B12 and folic acid, they are helpful for maturation of the RBC. Maturation of RBC. If maturation is not happening, what will happen? It will lead to megaloblastic anemia. Here the RBC size is bigger but its usage is less. So megaloblastic anemia. So these are the features that is required for the formation of hemoglobin. Out of this, the most important mineral is iron.